It is now my pleasure to introduce tonight's first award. Established in 1975, the William B. Coley Award for Distinguished Research in Basic and Tumor Immunology honors the legacy of Dr. Coley, a well-known New York surgeon of the 19th century who is now considered the father of cancer immunology, immunotherapy. Gosh, I'm so sorry. The one word you don't want to screw up, right? Anyway. His work was the first to link the power of the immune system to the treatment of inoperable tumors. I mean, it was like the first guy who looked at a lobster and was like, I'm going to eat that thing. I mean, these were like genius men. His work later inspired his daughter, Helen Coley Knotts, to found the Cancer Research Institute 59 years ago. Tonight's Coley Award winners, 5 in all, received CRI's top scientific honor for the seminal work in basic and tumor immunology. And if you think it's easy to nab one of these, think again. Let's just say that my B+ in Mr. Anacone's 9th grade chemistry class, the hardest class ever taken, by the way, still wouldn't come close to how much work these guys have done. So presenting the first 3 awards is Dr. James Allison. Sit back and let me tell you a little bit about Dr. Allison. He's the director of the CRI Scientific Advisory Council. He's the co-director of the CRI Cancer Immunotherapy Consortium and editor-in-chief of CRI's online scientific journal, Cancer Immunity. Yeah, his business card reads like War and Peace. He like carries it around in a suitcase. How much do you do? And that's just his volunteer gig. In his day job, he's the director of the Ludwig Center for Cancer Immunology. He's a Howard Hughes Medical Institute investigator and the David H. Koch Chair in Immunological Studies at Memorial Sloan Kettering Center. Hello, overachiever. Seriously. He also is a mean harmonica player. That is a true story. He's also a true Texas boy in every sense of the word. I am told that he loves his country blues. I am also told that he is known to sing the periodic table to the tune of Willie Nelson's Always on My Mind. Is that true? Because if that's true, there's an opening on Saturday Night Live, and they would love to have you. Mostly, though, and you and I and everyone here know this, he's known for the scientific stuff, mainly as a pioneer in the development of a new class of immunotherapies called checkpoint blockades. His anti-CTLA4 antibody received FDA approval last year for the treatment of advanced melanoma, the first therapy of any kind ever that was proven to extend the lives of patients with this disease. Also, by the way, anti-CTLA4 just won People Magazine's sexiest antibody. So they got that. No, it actually did win an award for best biotechnology project. But I just thought sexy antibody was so much more impressive. Dr. Allison received CRI's Coley Award back in 2005. Please help me welcome to the stage, Dr. Allison.
After all that, I'm a little disappointed I didn't bring my harmonicas with me. We could have really gotten down here and had some fun. But on a more serious note, I just want to say that, you know, as director of the Scientific Advisory Council, the Cancer Research Institute, really happy to be able to present the 2012 Coley Award to 3 of my colleagues and friends over many years. Richard Flavell, who unfortunately can't be with us, Lori Glimcher, and Ken Murphy. And without getting into a lot of science with you, I just want to tell you that one of the most fascinating things about the immune system is its complexity. There's a lot of different kinds of cells that go through your body and interact in very complicated ways and make hormone-like substances and figure out how to kill infectious agents, figure out how to kill tumor cells, protect you from viruses, but without destroying your normal tissues. And so we've known basically for a couple of decades that there were basically 2 kinds of lymphocytes that could do that. CD8 cells, which killed, and CD4 cells, which people didn't really realize how complex they were. But it began to be shown by a number of different labs that CD4 cells were really a complicated set of cells, that they made different things, that did different things in response to infections. And this is really important because you have to have the appropriate response to protect. Inappropriate responses can either be ineffective or as I mentioned earlier, result in more serious consequences by destruction of normal tissues that wouldn't have happened otherwise. And so there were these clues that there were this complexity in CD4 T cells, but it became, as it happens with scientists, it's like, why are there differences? How do we figure out what drives this? And so the three investigators that we're honoring tonight all made major contributions to understanding not just the complexity, but what drives the complexity of this component of the immune system. So basically, they all, in one way or another, showed that there were transcription factors, and I won't get into that except to say that transcription factors are things that sit on the DNA and determine which genes will actually, of your 30 to 50,000 genes, which one actually gets made into proteins, and they can program a cell to have a certain sort of response. And so in the early to mid-1990s, Ken Murphy, who's at Washington University School of Medicine in St. Louis, was one of the first to open the door to understanding the molecular clues that drive CD4 T cell differentiation when he used a model system that he developed. He found that the signaling molecule interleukin-12 was critical in driving the differentiation of what are called helper cell 1, or Th1 cells. And 2 years later, he showed that the transcription factor STAT4 was involved in the process. In 1997, Richard Flavell at Yale University showed that the transcription factor GATA3 was a critical regulator of the development of the other major kind of T cell, Th2 cells. It was the first time that a sole factor was shown to be necessary for T helper cell differentiation, and suggested that there were these master regulators that could actually tell a cell, you make these genes and you do this thing. And that's what you do. And finally, in 2000, Lori Glimcher, while at Harvard Medical School, identified the transcription factor Tbet as the master regulator of the Th1 class of T cells, which showed, again, there was this one thing that gets turned on that really drives all the things that happen after that and makes the cells do what they're supposed to do. So anyway, in the years between that work and the present, all three of these individuals have made additional discoveries that shed light not only these transcriptional pathways that mediate CD4 T cell development, but many other fundamental discoveries about the immune system that I could go on for a long time about, and to dessert probably, but I've been told I can't do that. I've got to stop in a moment. Anyway, together, these three people studies revolutionized our understanding of T cell biology and have given scientists important clues into how to intervene therapeutically and to achieve the most efficient immune responses to diverse threats, including cancer cells. So for their pioneering work to define the transcriptional factors that regulate CD4 T cell differentiation, it's my honor to present the 2012 Cancer Research Institute William B. Coley Award for distinguished research in basic immunology to Drs. Richard Flavell, Lori Glimcher, and Ken Murphy. And as I mentioned earlier, Dr. Flavell could not be with us tonight, but he expressed his regrets that he wouldn't be able to accompany us. And so first I'd like to ask Lori to come up and accept the award. And I should add that after nearly 30 years at Harvard, Lori joined us this year in New York, where she's now the dean of the Weill Cornell Medical College. So, congratulations.
Let me assure you all that Jim plays a mean harmonica. I've heard him many times. I'm really delighted to be receiving this award. I have been a long-time admirer of the Cancer Research Institute. They seem to me to embody the very best in their mission of funding and promoting basic and translational immunology, especially as it relates to cancer. It's even more gratifying to me to receive this award with two scientists who I have known for a long time and admired for a long time, Ken Murphy and Richard Flavell. The three of us have dedicated our careers towards understanding the regulation and function of lymphocytes in the immune system and how, and trying to exploit that knowledge to enhance human health. Science can often be a lonely affair, and the rewards are often private, so it really lifts up my spirits and those of the wonderful young men and women who have worked in my lab for the last 20 or 30 years, some of whom are here tonight, to receive an honor like this. And it is on their behalf, as much as on mine, that I accept with great pleasure the William B. Coley Award. Thank you so much.
And so I'd like to ask my good friend and guitar player, Ken Murphy, to come up and accept his award now as well.
You want me to say something?
Yeah, go ahead. Whatever you want. You got this.
Well, it's very nice to have received awards like, to receive an award like this, especially with Lori and Richard. Too bad Richard can't be here tonight, because the lab has been working on this area Jim talked about over the last 2 decades, since the early 90s, to try to understand at a basic level what drives T-cell immunity, which is, I think, the next major issue. So I accept this award on behalf of all the people in my lab who have actually done the work, and then the other people in my life who've made it possible. I especially want to thank some early trainees, Chi-Song Shea, Suzanne Zabo, Nils Jacobson, Sheila Ranganath, and Wenjin Ouyang for their early work on T-cell development. And especially I'd point out that Suzanne went from working on IL-12 and STAT4 in my lab to Lori's lab, where she did the next best thing, cloning T-BAD, which, of course, is a major discovery. And I'd like to acknowledge Ann O'Gara, a collaborator who helped in the early work on T-cell development. I'd like to thank some close colleagues, Casey Weaver and Bill Shaw and Andre Shaw, who's in the audience tonight, for freely sharing lots of good ideas over the years, which we've stolen and used as best we can. I need to thank Emil Unanaway, Bob Schreiber, and Paul Allen for forging the immunology community at Washington University, which is now involved in the Siteman Cancer Center's tumor immunology program under Bob's guidance. And that's a major development for us at Washington University. I should thank my 4 sons, my 2 parents, Paul and Lorota, my 1 brother and 3 sisters, for their encouragement and guidance over the many years. And there's one person who falls into 2 categories, not just having made the work possible, but also doing the work, and that's Dr. Teresa Murphy, who's in the audience tonight, for allowing me to work side by side with her since the very beginnings of the Murphy Lab. If we cloned it, expressed it, knocked it out, or sent it to you, it was probably her, not me, who did it, and there is a relation. I am her husband. So I accept this award on behalf of those people who did the work and made it possible. Thank you.